going to talk about immunity. So immunity is one of those areas of medical science which is, a lot of it is actually known, a lot of it is actually also not known. And it is really becoming across. It is going to be a long class, but immunity is a really interesting topic. In your day-to-day life, there are lots of examples where the immune system is your savior for all of us. So it is fascinating to know how these things work, right? And also, we will see a lot of relatable things and applications. Let me just give you two examples. So, round the year, we have always dealt with fever, sometimes or the other. At least once or twice a year, we always we suffer from fever. So, fever is a response of the body to an infection. Right? Of course, which all we there are other causes also, non-infective causes, but we learn them in later years. So, why is it that the temperature rises in the body? Does it help the body in any way when it is invaded by pathogens? Right? So, we'll see trying to answer this. Increase in temperature specifically, does it help the body in any way? Second experience, all of you had a lot of vaccinations as you grow up. Right? And so vaccination works on a very important principle of acquired immunity. So what we do in the process of immunization, vaccination, the latest one that all of us went through was of course the COVID vaccines. Right? So how is it supposed to act? We are sensitizing the body to low doses of antigen, and we expect that the body remembers this, and whenever there is a fresh attack, a natural infection, the body should respond to it with full vigor and full force. So that is what we expected with the premise of immunization, right? So this is why, these are some examples of why we should learn about immunity. So when we try to define immunity, then there are some terms like antigen, antibodies, epitopes. We'll differentiate into the different types of immunity, right? Classification. And thirdly, we will try to see or understand the mechanisms of innate and adaptive immunity. Immunity. So it is the capacity or ability of the body to defend the body or respond to. There are 2-3 things. Generally, we have the idea of it is only going to respond to pathogens. Pathogens are organisms which cause disease or pathogenesis. So pathogens, but it is also the ability of the body to respond to antigens. Now, antigens will evoke an immune response, but they may or may not cause disease. Right? So all pathogens, because they are provoking the immune system, they are triggering the immune response and causing disease, they are on all antigens. All antigens need not be pathogens. So pathogens, antigens, and thirdly, that I think it's new to you, that immune system also has a very vital role in defending the body against development of cancers. So it is also acting against tumor cells, right? So coming to these terms again, antigen, it is a substance, any substance that triggers the immune system and produces an immune response. Now, epitope is a specific part or area on the antigen which can be recognized by the immune system and which can produce a response. Right? So, one antigen or one, say think of it as a single bacteria, for example. So, one bacteria can have multiple epitopes, meaning one bacteria, the bacterial antigen can have multiple molecular patterns on their surface, each of which can be recognized by our immune cells, the B cells or the T cells, and each of which can be responded to by an immune response. So this is what we call as an epitope. So a single antigen can have multiple epitopes, right? And third one is antibody. So antibodies are proteins which can bind to antigens, right? So antigen binding proteins, and which are secreted by plasma cells. So plasma cells are cells which are activated B cells or B lymphocytes. So these are some basic terms to start with. So all of you are aware that there is a 3-tier defense system of the body, right? So we'll try to put them in perspective of some organization. So the first line of defense, usually we talk of the skin, intact skin, mucous membranes, right, and their secretions. And how they act, what are the contents in the secretions that is enabling that, we'll come to it later on. And normal flora. So what is flora? Flora is, when we call as, what we refer to here as normal flora is the bacterial microenvironment, right? So your body normally has lot of colonies of bacteria and other microorganisms which is normally there, right? We call them as commensals. So this is also taking part, surprisingly, the bacteria itself is taking part in the defense system of the body, right? Second line may we have the different innate immune cells, the granulocytes, inflammatory response of the body, complement system of the body, and some antimicrobial proteins which are produced by the cells. So this is the second line defense. And third line defense is the specific cellular lymphocytes, B and T lymphocytes. We classify them, the first and second one, these are non-specific responses, meaning against any organism that enters the body, these responses or their defense mechanisms are same. It is not specific to the type of bacteria or the type of organism, right? Adaptive immune system is a specific one. So third line defense is a highly specific way of responding, right? And this is what we call as adaptive immunity. So first and second come under natural or innate immunity, and third comes under acquired or adaptive immunity. Moving on to the classification. So what we call is innate and what is adaptive or acquired. Anyone? This basic ideas you already have from your high school knowledge, right? So innate का मतलब क्या है? It's something which is, so the person is having it by the virtue of his or her birth. From birth it's there, right? So this is innate or natural immunity. Adaptive is something which we gain in the course of exposure after birth. So hence the name acquired. Now both innate and adaptive immune systems, they have humoral components and cell-mediated components, right? So this should be clear that humoral and cellular immunity, these are components which are present both in innate as well as adaptive, right? It should be very clear here because in various books you will find there is some confusion regarding, they describe it in various ways. Humoral ka matlab hai, it's something which is soluble proteins, right? And cellular means specifically the cells have something which can trigger a protective mechanism. We also classify immunity into active and passive, right? Based on the way they have been gained, right? So active immunity can again be gained naturally or artificially. So every natural infection that we have, it stays in the memory of our adaptive immune system, right? As memory cells, memory B and T cells. So this is a form of naturally acquired active immunity, right? Artificial ones, vaccinations. So vaccination, it is a procedure that is done to produce some form of memory. So this is again an active immunity because we are not giving ready-made, say, granules, or we are not giving ready-made antibodies. We are giving the dead antigen or a weakened antigen. You are introducing that to the body with the understanding that because it is either dead or it is weakened, it is not going to produce a very strong response, right? Because what is otherwise, kya wajah ki why do we want to deliberately introduce an infection and see? So because it is weakened or either it is dead or inactivated in some way, it is not producing a immune havoc or immune response in the body, but the low level of stimulation stays in the memory cells. So that is what we have in vaccination. So it is an artificially acquired active immunity. Passive immunity में we are giving ready-made forms of mechanisms हम लोग directly दे रहे हैं. So that is why the name passive. So during pregnancy and after pregnancy also, during the process of lactation, immunoglobulins pass from the mother's blood to the baby, pregnant fetus, and also in breast milk we have lot of immunoglobulins. So ready-made immunoglobulins or antibodies are transferred to the baby now. So this is a form of passively acquired immunity, but it is a natural process. And this one which you see, you can artificially produce some immunity or some immunoglobulins artificially, right? So monoclonal antibody techniques, you read about the technique details in biochemistry. Specific type of immunoglobulins can be manufactured and these are used also. Tetanus के against जो immunoglobulin दिया जाता है, rabies के against the immunoglobulins, these are all ready-made immunoglobulins which are administered in different situations. So these are the aspects of active and passive immunity. So if you get a question on classifying immunity in your exam, depending on the marks assigned, we should talk about all these aspects. Okay. So can anyone read through what are the properties I'm trying to depict here in these pictures? So let's say this is the easy one. So anyone, this is the memory cell telling to a bacteria Escherichia coli. So what property is this? Louder, anyone? Memory, right? So immunological memory. This is one good. Easy tha. Baki dono cheeze can you interpret in your own way. This one. Fingerprints are something more specific, and also so for each individual it is specific. So it is a very diverse repertoire. So this is why this picture. So we are here I'm talking of diversity. And the last one is specificity. So these are the three key attributes of acquired or adaptive system which is not present in the innate system. So let me try to differentiate. So innate and adaptive, it's a very common concept and also a very common question. So can both these mechanisms differentiate between self and non-self? कि कौन सा टिश्यू या कौन सा प्रोटीन बॉडी का अपना है या कौन सा अपना नहीं है? So can it differentiate between that? Yes. Both innate and adaptive can differentiate. Lag phase. So lag phase is the time from the time of exposure to the response. So innate immunity में lag phase is absent, right? And in adaptive it is present. Why? Can anyone answer why? Why? Because it is non-specific, of course, and so this you will see in the coming slides that the components whichever are there. So for them to produce a response, we do not need any type of activation. As it is here, the moment they come across something, they will provide their own response. So lag phase is this. Third aspect is specificity. So innate immune system specificity is very limited, right? As I said, why is it limited? Anyone? Specific innate system is the naturally thing which is already present. So कुछ basic चीज़ें हैं कि it can recognize what is a prokaryote, what is a eukaryote. So very basic patterns of recognition are present, right? But because it is not the system which the person is gaining in the course of exposure, so specificity is bound to be limited here. So here it is highly specific, and that's why highly diverse. Okay. So diversity point also I have covered. And lastly, memory we all understand. T cells or B cells, so the mechanisms are always they are having a lot of immunological memory. That is the sole premise of the adaptive system, the vaccines, right? For the innate immune system, this is absent. So that's why कैसा भी exposure हो, the response is always going to be the same. So these are the important differences between innate and adaptive. We also have the concept of primary and secondary response. So adaptive immune system में whenever there is a first exposure, so this is the time, suppose it's a new exposure, new antigen. So some time is spent in identifying, and of course there is a response. So this is the magnitude of the response, which you can measure in, for example, in B cell responses we can measure in terms of the antibody titer, serum antibody titer, कितना antibody बना है. Same exposure antigen X, if there is a second exposure, so second exposure happens without lag phase, a very minimal lag phase, and it is a highly magnified response. So this response is called as a secondary response. So because memory cells already know which is the organism, so there is no time spent in recognition and identifying, and there is a huge response. Serum antibody titers, there is a huge magnified response. This is called as a secondary response. Now let us come to the what are the different players or the soldiers which are present here. So we'll go with innate first and then adaptive. The innate immune system में the components which are participating, these are the barriers of the body, anatomical, physiological and inflammatory barriers. Secondly, the cellular barriers of soldiers, so phagocytes and natural killer cells, and humoral is the third one, which is a soluble thing. So complement system and interferons, these are the soluble components of your innate immune system, right? Adaptive में five go. So adaptive specifically is the variety बहुत ज़्यादा नहीं है, it is just the B and T lymphocytes. The variety is there in the receptors. So B lymphocytes, they form plasma cells when activated. This is one. T lymphocytes. Now there are various types of T lymphocytes. Now CD8, CD4 and CD25. What are this? So CD is a short form for cluster of differentiation. So these are some surface proteins which are present on the surface of T lymphocytes, a specific subset, and hence the name. इस group में और इस subset में this specific type of CD protein is present. So hence the name CD8. So this numbering has been, of course, the course of discovery they have been numbered, right? So CD4 proteins जिनमें present है, we call them CD4 positive T lymphocytes. CD8 जिनमें present है, CD8 positive, CD25 likewise. So CD8 positive T cells, these are cytotoxic T cells. That means they can directly kill a cell. Hence the name cytotoxic. Right? CD4 positive cells are helper cells. They are further subdivided into helper 1 and helper 2. So this helper 1 and helper 2 again, they help different aspects of the immune system. Hence the differentiation TH1, TH2. And CD25 are the ones which keep everything in balance. The immune response ज़्यादा ना हो जाए. So it is a balanced thing. So suppressor or regulatory T cells. These are the different soldiers now. No? So basic scheme of the immune response is this. Right? So as soon as antigen enters, right, a pathogen enters, initial response is by the innate system. Right? So innate system में जितने भी चीज़ें हैं, phagocytes हैं, antibodies, soluble components, this will all act. In the meantime, the message is also conveyed to the adaptive system. Right? So antigen-presenting cells, specific cells, as the name tells you, इनका काम बस ये है कि antigen को पहचानो and present it to the adaptive immune system. So इसी दौरान these cells are present in different portals of the body, skin में, right, GI tract में, respiratory tract में, everywhere जहाँ पे antigen entry का एक portal बन सकता है. So in all these places we have this sentinels which guard. So antigen presenting cells, they also phagocytose the antigen, but their basic purpose is कि उसको मतलब अंदर लेके फिर उसको they will process it in some way. And specific antigenic peptides, right, corresponding to say epitopes, they are presenting them to the T cell or the B cell in the lymph nodes. Why? Because इनका जो response है T cell या B cell का response है, that is going to be very specific, and it will also stay in the body. The memory of that will stay, so that अगली बार के लिए body can be prepared. So all of this is happening with each time. Okay, now if the B cell is activated, we have the plasma cell which secretes antibody, and if the T cell is activated, we have the different forms of T cell response that we just saw, either this one or this one. Right? Now, why in some cases now B cell response will be there, why T cell, this will come up in the course of discussion. So I will go directly to the mechanisms now. Now for any mechanism to be very effective, immune response एकदम effective हो, तो दो steps है उसमें. First step is recognition, चाहे वो innate का हो या फिर adaptive का. For both of this, recognition पहला step है. Once it is recognized, so then there is a response proper. Now innate immunity में जो response recognition के mechanisms present हैं, which are the two things. Molecular patterns of pathogens, these are called as PAMP, right? So course of evolution में for all the prokaryotic and maybe the beginning forms of other organisms, basic molecular patterns on their cell membrane or cell wall, these are constant across species. Species to species variations हैं, कुछ उसमें ऐसे patterns हैं which are very constant, right? The basic patterns. So these are say two examples I've given here. Lipopolysaccharide, right? It's a part of the bacterial cell wall, right? Mannose, एक और component है in cell membranes and cell walls which can be recognized by different aspects of the immune system, different cells of the immune system. So these PAMPs are recognized by the receptors of the innate system. So these are the receptors are pattern recognition receptors, PRR. So some of them can be soluble, say NBL, mannose binding lectin, LPS, lipopolysaccharide binding protein. So these are the two ones. And it can also be cell membrane bound, toll-like receptor. So ये दो receptors ऐसे हैं which are recognizing or participating in innate immunity. And as I have emphasized it again and again, so these are some basic patterns which are present across all forms of prokaryotes. ताकि as soon as they enter the body, they are recognized by the human immune system, which is a very sophisticated. Adaptive immune system में recognition is done by receptors which are called as somatically generated receptors, SGR. These receptors develop by the process of gene rearrangement as the body gets exposed to more and more antigens. And hence the specificity. The gene rearrangement is the process which is enabling the presence of huge amount of diversity, specificity, all of this. Now B cells, they recognize soluble antigens. This is important. When this antigen binds to the membrane bound antibody. So the B cell receptor is a membrane bound antibody or immunoglobulin itself. T cell receptors can identify or recognize peptides, that means fragments of the antigen when they are combined with MHC molecules. Now here you come across a new term, MHC. I'm coming to this in a bit. And MHC molecules kahan pe? So when MHC molecules which are present on the surface of antigen presenting cells. So APC we have just seen in the previous slide. कि there are some cells जिनका purpose ही है कि they will process and present antigen कि लोग खाओ अभी, right? So these APCs they have certain MHC molecules, right? So they present it to the T cell, right? So this is how a T cell can be activated. So it cannot be activated in some other way. Okay. Now what is this MHC? So MHC basically is a cluster of genes which are present in chromosome 6. And the products of these genes, they take part, look at it, like intercellular recognition. So these glycoproteins, they are participating in recognition and discrimination between self and non-self. So this is what it does. So MHC एक gene का cluster है. Now there are three forms of MHC that we know, which are expressed. MHC1, it is expressed in the surface of all nucleated cells and platelets, right? So will a RBC have MHC1? RBC? Will it have? So mature RBC does not have a nucleus, right? MHC2. So MHC2, it is present specifically on the surface of antigen presenting cells. So macrophages, they function not only as phagocytes but also as APCs. Dendritic cells and B cells, they also function as APCs. So from these two statements, the point to be really read in between the lines is MHC2 is present on the surface of macrophage, the APCs, which are also nucleated. So antigen presenting cells, they have both, because they are nucleated cells, they will also have MHC1 and additionally they have MHC2, right? MHC3 are components, so they are expressed on the components of complement system. Complement system, will come to this in a bit. So these are the three types of MHCs, classical MHCs that participate in immune recognition. So this is how it looks like. So MHC1 and MHC2, as I said, cell membrane bound glycoprotein cell. So this is the membrane bound part and this is the part which is projecting outside. Now you have here alpha and beta chains here, and in between you would have the antigen binding clefts. So cell ke andar, once the antigen is processed inside the cell, right? So small fragments will be presented to the T cells in this antigen binding or peptide binding clefts. So chote parts bound in these clefts, it is presented to the T cells, right? MHC1 and MHC2. So we do see that there is a difference, right? MHC1 mein, the peptide binding cleft is in the alpha chain itself. MHC2 mein, it is between the alpha and the beta. The small differences हैं. ये चीज़ें second year में और detail में you learn in microbiology. Now what are these APCs, antigen presenting cells? As I said, these cells exist solely for the purpose of immune recognition. So which presents the fragments of antigens to T cells. We broadly classify them as professional and non-professional ones. Professional ones are those which always have this function. Dendritic cells present in the skin, macrophages, B cells. So they always function as antigen presenting cells. Non-professional ones are those which will function as APCs, not as a primary function, but they can function when required and they function at some phases of life. Say for example, thymic epithelial cells. These are epithelial cells of the thymus gland of the body. So these cells function as APCs when the WBCs are in the developmental stage. T lymphocytes जब develop हो रहा है, so in that phase they function as antigen presenting cells, right? So hence the name non-professional APCs. Okay. So this is a structure of the B cell and the T cell receptors. So remember, adaptive immune system में दोनों चीज़ important है. तो MHC कहाँ पे present है? APC में. And T cell and B cell are actually the areas where they will go and present it too. So MHC bound antigens are presented to the T cell receptor. T cell में this is the structure of the receptor and specifically this is the receptor proper. These other peptide chains that you see, these are also part of the receptor complex. But recognition may, this is the part which is important. You can see the name antigen binding site. And T cell receptors are self-MHC restricted. So this is a very important statement. What does it mean? कि अगर antigen MHC में नहीं है, so T cell cannot recognize it. So this is the importance of this statement. Self-MHC restricted. B cell receptor where you try to understand the picture here. So these are what? Immunoglobulins. Right? You have read about immunoglobulins in biochemistry already. So you have some idea. So B cells में immunoglobulin chains themselves serve as receptors. So when these chains are cross-linked by an antigen, as you can see here, so cross-linking will trigger the recognition. Right? So primary functions or signals are the cross-linking, but you learn in the later years कि in each of this recognition, primary के साथ there is also a small co-stimulatory signal which is present, दोनों में, right? So signal 1, signal 2 that you can see here. So why is it important? These things are important for maintaining a regulation of the immune response. Like it should not just randomly get activated. That's why that's one more level of activation required. Right? So as soon as it is recognized, this is the first signal, and the additional signal is one layer of protection. Okay, now moving to the mechanisms. So mechanisms of the innate immune system. So you already know the granulocytes. You have read about that in the last theory class. You have also seen these cells under microscope last week, right, in your peripheral smear. So granulocytes mean the contents of the granules. What are this? If anyone can answer. Contents of the granules. We have some strong enzymes, right? Enzymes which can perform some form of intracellular killing in the event of a phagocytosis. These are strong digestive enzymes. So before we move on to this, again step by step we'll go. So innate immune system mechanism में तो पहला जो चीज़ है that is the barrier systems of the body, right? So when we talk of barriers, skin we understand. Intact skin is the biggest barrier that we have. If the skin is intact, most organisms will not be able to enter. So this is a mechanical barrier. Acidic pH. The skin also, the pH of the skin also has some, immune effect, and sweat. Sweat also takes part, like it is also functioning as one level of barrier. So these are three reasons why, or three ways how the skin is acting as a barrier. Mucous membranes. So mucous membrane में mucus itself, it can, because it's a very sticky substance, so most organisms will get trapped in the mucus. So mucus is a barrier. Now is it sufficient just to have mucus? So it is not sufficient. So the mucus, it needs to be expelled out of the body at regular intervals. Otherwise what will happen? Suppose in all mucosal surfaces we have this mucus secretion. Think of it, respiratory tract में mucus है, बस बहुत mucus है, and उसमें there are lot of organisms which are trapped. So if these are not expelled, the बहुत देर अगर वो रहा वैसे तो this will serve as sources or nidus of infection. Right? So hence, in all these areas you will also see the epithelial lining is a ciliated epithelium. So what is, so you see the beauty, how things are present in the body. Ciliated epithelium, when the cilia beat, the direction is always to expel it from outside, from inside to the outside. Together, these two things are called as mucociliary clearance. Mucus and the beating action of the cilia which expels it. Okay? I was talking about the normal flora. What is that? So in various parts of the body, there are some microorganisms which are normally present. वो आके रहते हैं वो, right? So they have a sort of a symbiotic association with the body, right? So because वो वहाँ पे normally present है, so they will not allow any other organism to enter. So that is how they function as a barrier. These are called as commensals. So one of the reasons we avoid indiscriminate use of antibiotics is this: कि antimicrobial or substances, antimicrobial drugs, when we take, so it might also end up affecting the commensal flora, right? So एक immune system, एक immune portal we are actually harming if we use antibiotics indiscriminately. So do you have a concept of gram-positive and gram-negative organisms? Do you have this idea? So suppose we know for sure that this antibiotic X is effective against gram-positive organisms and not against gram-negative organisms, right? तो ये हमें पता है. But what we see is many times we are not bothering to actually go into and identify what is the exact organism. And empirically, we end up prescribing a lot of broad spectrum antibiotics. So, which in a lot of cases, it ends up also harming the commensal layer, commensal bacterial layer. So, this is the importance of this point. Now, moving on to the physiological barriers, right? What is that? Temperature. So, if the temperature increases, so enzyme activity will reduce. You have read about this in school, right? So, whenever we get invaded by organism, the immune response that starts, it leads to a release of a lot of cytokines, interleukins. So, one of the interleukins is interleukin 1, the second one is interleukin 6, and tumor necrosis factor alpha. So, three ऐसे chemicals हैं which is released in this process. So, these chemicals through the bloodstream they reach the hypothalamus, and in the hypothalamus they affect the temperature regulating system of the body. Because of this affect the temperature, the body feels that as if suddenly the temperature is, the body has become cold, so it triggers heat generating mechanisms of the body, shivering, right? And shivering is a form of muscle activity, so muscle activity is always exothermic, so it adds to heat production in the body. So this is how fever is happening. And once the temperature is risen, it has a bacteriostatic effect. So it restricts the enzyme activities in the bacteria, affecting their replication. So this is how fever is helping us actually. And of course, because the temperature is risen, so it is also a very important symptom and a sign which will alert the person. The stomach and vaginal secretions. So two areas I have given some examples. So these are also very strong forms of defense. What is the pH of the gastric acid? Stomach में जो secretions हैं, any idea? Acidic है, we all know that in stomach में there is HCl. This is, you know this from our knowledge of school. But can you tell me the idea of what is the pH? 1 to 1 point. Yes, very good. So it's in the range of 1 to 2. So think about it. pH of 1 to 2 is a very strong acid. अगर इस acid में 1 to 2 pH में if you put your hand, you will sustain a severe acid burn. It is that severe, but we don't realize actually, right? So this is a very strong acid which is protecting the body from a lot of things we eat. Lysozymes. These are hydrolytic enzymes present in secretions. Two examples, कहाँ पे present है? Tears. These are present. Saliva. These are present. These secretions additionally, tears and saliva also have some immunoglobulins in addition. We come to this. Acute phase reactants. So acute phase reactants refers to a lot of proteins. Common of them being the CRP. So if you come across a report, a blood report of a patient admitted and or OPD basis पे who is getting treated, you will invariably come across this CRP level itna hai. So what is CRP? It is actually acute phase reactant. CRP naam aa raha hai from the C polysaccharide component of the bacteria. So this protein, it can bind to the C polysaccharide component of the cell wall and activate the complement system. Hence the name C reactive protein. So you will invariably come across this point CRP in the blood reports or investigation reports of any patient who is suffering from infection. This is a very commonly advised investigation. Commensals, I have already talked about this. Moving on to the cells now. So we have talked about the barriers, remember. Secondly, we are coming to the cells. So granulocytes, they function as phagocytes in the body, right? So phagocytosis process, अगर आपको बोला, what is the process? It's the process of, anyone who can define what is phagocytosis? Engulfing. It's the process of cell eating in a very simplistic way. So the cell, in this case the granulocyte, will give some projections, it will encircle it and engulf it. Once it is inside, what happens? So it is inside the phagosome. The cells also have lot of digestive enzymes, the lytic enzymes. So phagosome fuses with the lysosome, the hydrolytic enzymes which are there, and forms a phagolysosome. जहाँ पे within the cell things are broken down, right? And after everything is digested and after everything is broken down completely, the residual matter is extruded out of the cell. So these are the basic processes of phagocytosis steps. Now macrophages and neutrophils, these are two important phagocytes that we have. Why did I mention macrophage earlier? Macrophage is a WBC, it's a phagocyte which is derived from which cell? Monocytes. So monocytes, they spend some hours in the blood. After that, they migrate to the tissues of the body. Their size increases, their antimicrobial potency increases, and then they are known as macrophages. Right? Macro means large. Phage comes from the word phagocytosis. So they can actually engulf hundreds and hundreds of bacteria compared to a neutrophil which could engulf only say in terms of 5 to 10. So that's why they need macrophage, and neutrophils are also sometimes referred to as microphages. So within the tissues in different parts of the body, monocytes, now known as macrophages, guard the openings. Say in the liver, they get a specific name, the Kupffer cells of the liver. Right? Within CNS or brain tissue, उनका नाम हो जाता है microglia. Within the respiratory tract, they have the name pulmonary alveolar macrophages, PAM. Right? So these are just some examples of what we call as the monocyte macrophage system. Right? So this is why I mentioned macrophages earlier, because as soon as the organism enters, it will actually come across this one earlier, right? Of course, neutrophils are already there. They are the ones which respond the first, right? So neutrophils, once they are able to deal, it's fine, done and dusted. If they are not able to deal, the chemicals or cytokines which are again released in the process of this immune reaction, they will activate the macrophages, right? That's why they act in concert. Okay. So down here what you have seen is the process by which neutrophils and monocytes they react. So first is the step of margination. Blood cell में वो पहले बीच में चल रहे थे. So now first thing they will come towards the side. This is the margination process. Diabetes, they navigate their way through the endothelial cells. Capillary endothelial cells के बीच में से squeeze करते हुए somehow they enter the tissues. And chemotaxis. Chemotaxis is the organism themselves, and या फिर जो पहला reaction है, chemicals they attract the phagocytes. Chemotaxis, and then the process of phagocytosis proper, right? Okay. So they also release these chemicals, as I said. So chemicals, इनका तो function already I have told you. IL-1, IL-6, TNF alpha, they also react. They also produce fever. तो एक फंक्शन उसका ये है. These cytokines also serve as chemotactic factors जो बुला रहे हैं बाकी phagocytes को. And look at this one. This is a very fascinating thing to me. GM-CSF. GM-CSF is a hemopoietic growth factor. Granulocyte monocyte colony stimulating factor. तो ये cells इस immune pattern में they are also releasing some factors which will reach the bone marrow, and bone marrow को कहेगा कि you need to produce more and more cells. So a signal also goes to the bone marrow. So this is the function of the phagocytes. Right? You covered all of these aspects. Right? If you get this question in exam, phagocytes, it's not only phagocytosis, but other things are also there. So just illustration of the process of phagocytosis. Just skip through. Yes. Now, intracellular killing. What are the ways by which you can, the phagocyte or it is able to kill? Reactive oxygen species is a very important one which is able to neutralize the organism inside the cell, right? Hypochloracecine, lysozymes and proteases, nitric oxide defenses. All of this, these are all there in the book. You read the contents of the granulocytes, these are all there. So all of this participate in the intracellular killing. Now natural killer cells. So we are talking of mechanisms of the innate. We've talked about barrier, and now we are talking of cellular. Cellular, we have talked about phagocytes. Now we are talking of natural killer cells. So this natural killer cells were first identified in 1976. So it's a fairly recent set of cells that we know. Now lymphocytes, it was observed that around 5 to 10% of the circulating lymphocytes, they're slightly larger and they have granules, which is unusual for a lymphocyte. Because lymphocytes we always classify them as agranulocytes, right? But this, so around 5% of the lymphocytes, they have granules, and these are the ones we call as natural killer cells. Natural killer cells, they look for altered self cells. So this is very important. So if the cells are normal, cell membranes are normal, membrane में कुछ extra protein या antigen express नहीं हुआ है, so it will not react, right? But the moment the surface molecules of the self cells are disturbed, so then they become active. So why? Because natural killer cells, NK cells in a short form, they have receptors for MHC1, right? MHC, you remember MHCs? So MHC1 ke receptors present hai, and you see they also have some activating ligands. So activation ke all mechanisms being present. So normal cells, nucleated cells and platelets, they have MHC1, and antigen presenting cell उनमें MHC1 भी है, 2 भी है. अगर सब कुछ normal रहा, so they will not respond. So normal cells with a normal MHC class 1, if that is present, they produce inhibitory signal. So these are circulating lymphocytes. Even if it comes across a cell, this may MHC hai, but MHC normal, so it will inhibit. This is an inhibitory interaction. But the moment where the surface MHC1 molecules are altered in the sense that structurally they have become different, or they have some antigenic peptides sitting on them, so then they are activating the natural killer cells. So ये important है कि altered MHC1 should be recognized for the NK cell to act. How they act? They produce perforins and granzymes. So small pores, perforins, these are all proteins. So small pores are inserted on the organism. Once the proper pore is formed, granzymes are introduced in the process of this interaction. Granzymes are enzymes which can break down the nuclear material of the affected cell. So this is how the natural killer cell is acting. So once natural killer cell ने उसको खत्म कर दिया, now it becomes a dead cell. The invading organism or the pathogen is now a dead cell. A dead cell which is almost equal to a tissue debris. So this is then phagocytosed by the macrophages and disposed of. So this is how natural killer cells act. Okay. So this is an example illustration of how a natural killer cell is acting. So moving on to the humoral components of the immune system. Humoral components may we have complement and we have interferons, two things. We are still in the innate immune system, remember, right? The complement system is a system of proteins which are present in the body, B, C, D, and these have various subdivisions. So whenever the complement system is activated, either it can be activated by an antigen-antibody complex. Complement can also be activated by mannose binding lectin, right? And also by a molecule called as properdin. तो किसी भी तरीके से वो complement system अगर activate हो, so it will keep on activating its components in a cascade, just like the blood clotting factors. पढ़ा होगा आपने intrinsic distinct school में, तो वैसे ही the things are activated one by one. The details of the complement system you will read in second year, but I am just giving an idea. Okay, this is also formed. So they are split up into components A and components B, and finally all this together they form a structure called as membrane attack complex, MAC. So this membrane attack complex, again you can see C5b, C6, 7, 8 and 9. What they do is, they attack the membrane, of course, as the name tells you, and they make these pores in the cell membrane, the cell wall. Through this, water can enter inside. क्योंकि natural killer cells में perforins के साथ the second thing that the cell itself introduced inside was granzymes. वो यहाँ पे नहीं हो रहा है. पहला step, it is almost similar. It is inserting pores with the help of this MAC. But then from the fluid enters the organism and it keeps on swelling up and eventually ruptures. So this is how complement is acting. ये mechanism भी non-specific है, right? So that's why it's a part of innate immune system. जो उसके बाकी components हैं, they can, जैसे कि आधे components they will form the MAC. So हर एक step में एक component split होता जा रहा है. This will function as opsonins. So what is that? Do you know what is opsonization? Have you heard of the word? I will tell you one. So let me show you a picture. So do you find it tasty? Yes? Someone is saying no? So imagine something different. Okay, so how can you make it more tastier? Sauce. I spent a lot of time in the north, so I remember hari chutney or lal chutney. Right? It makes it tastier. So this is what opsonization is about. Right? So if you are just having it like that, phagocytosis ho raha hai, right? But if it is coated with some chutney or sauce, this becomes more tastier. So these are the C3a and C3b. These are the which one is functioning as opsonins. And this process is then called as opsonization. So look at the scientific one now. So phagocytes are able to recognize antigens better when they are covered with these points. Interferons. You read about a lot of it during the COVID times, I suppose. So interferons are proteins which are secreted by cells in response to what? In response to viral infections. So this is a cell which has been infected by a virus. So it is releasing this purple cell, which you see in the purple color here. So these are proteins which are released by a virally infected cell. It is diffusing into the neighboring cells, and within the neighboring cells, it is activating antiviral proteins of the genome. So our genome has certain genetic sequences which are antiviral in nature. Kitna effective hai wo, that is a different thing to discuss, but wo cheez, we have it in our genome. But that needs to be activated. So and interferons, they go to the surrounding cells around the infected cell and they switch on these antiviral genes. So antiviral proteins are produced. These proteins are again nothing but enzymes which can break down DNA. So the moment the virus can enter and it tries to involve the genetic machinery, the material is broken down already. So a virally infected cell ke surrounding mein you have a shield now, right? So this is how interferons function. So this is about innate immunity, right? So can anyone summarize the points of the innate immunity? Why are you calling it innate? Because these are all present by birth and these are all non-specific things. Non-specific meaning for all organisms the response is the same, right? So mechanisms of agar pucha jaye, you will start with barriers, right? Anatomical, physiological, okay, and then you move on to cellular components. Cellular components mein we will talk about phagocytes, we will talk about natural killer cells, right? Okay, and moving on to humoral components or humoral elements, we will talk about complement system and we will talk about interferons, right? So we'll resume our discussion, right? So we learned about the mechanisms of innate. Now we learn about the mechanisms of adaptive immune system. The adaptive immune system is unique in the fact that it is very unique, right? Very diverse, and it retains, right? So whatever experience it has, it remembers that. It holds a grudge. Right? Okay. So two ways by which adaptive immune cells, right, can fight. It can either go to the cell and, like with a sword, it can stab the cell. One way of the battle is this. The other way is it can stand at a distance and it can fire something, right? Bullets, pellets, anything. The T cell acts like this, hence the name cytotoxic response, directly going to the cell and delivering how it is going to act. B cell acts from a distance, it shoots antibodies or immunoglobulins. Okay, so let us see what are the mechanisms exactly. So response mechanism we can differentiate into T cell and B cell response. T cell response we can further classify into cytotoxicity. When we talked about the cells, you have some idea. CD8 positive T cells, helper cells, right? Th1 and Th2, and regulatory cells. All of these are important. B cell mechanisms may we have neutralization, opsonization, complement, and ADCC. So ADCC is a short form for antibody dependent cytotoxicity, cell mediated cytotoxicity, ADCC. Okay, so recall this picture again. So when an organism enters, तो पहले तो कि innate immune system के जो भी वे मतलब players है या soldiers है, they are already there. But उसी time पे it is also trying to conveying the message to the adaptive system. How is that happening? Same time पे some of the APCs जो कि they are guarding the sentinels or portals of entry. इस example में this is the skin and this particular cell is the Langerhans cells. These are dendritic cells of the epidermis. So it is an APC, antigen-presenting cell. So it engulfs or phagocytoses the antigen, processes it inside, right, and presents it on the surface of MHC molecules. So I'm again drawing your attention to this basic scheme. Okay, adaptive immune system, the T cells specifically cannot function without MHCs. So it has to be presented and presented properly. The processing happens within the APC and you can see, so these are the MHC molecules which are holding the antigen and it is presenting it to the T cell. Lekin T cell kahan pe hai? So it will eventually reach the lymph nodes where the T cells are present and then it is going to present it to the T cells there, right? Developmentally, if I say, lymphocytes, they are localized to the lymph nodes, hence the name lymphocytes. Developmentally, after their phase in the bone marrow, B cell development continues in the same place, bone marrow, and T cell development, it continues further in the thymus gland, hence the name T cell. It is in relation to thymus, right? So whether an antigenic peptide, rather processing के बाद जो peptide है, whether it is going to be presented with MHC1 or MHC2, so that is actually dictated by the nature of the organism. So that is the irony कि organism खुद choose कर रहा है कि वो किस तरीके से मारा जाए, right? MHC1 या MHC2. So intracellular organisms like viruses, right? Some bacteria, not all, they are processed with a different processing pathway. Details I'm not going right now because you'll be reading the details in second year. They are presented through MHC1. And MHC1, it is going to present it to CD8 positive T cells. So 8 again is the product here. जो extracellular antigens हैं, most bacteria, product toxins for example, antigens, extracellular ones which are soluble within the blood or the body fluids, they are presented with the help of MHC2. And with MHC2, they present it to CD4 positive T lymphocytes. So यहाँ पे भी, if you take out the numerical values, it is again 4 into 2 equal to 8. And in the first case, 8 into 1 equal to 8. So this is how you can remember. So intracellular ones presented by MHC1, 2 CD8 positive T cells. Extracellular antigens presented by MHC2, 2 CD4 positive T cells. So remember, this part is the crucial recognition part. And क्योंकि adaptive system is a very highly specific system, if recognition is not there, it is not going to mount the response. This is very important. Now, cytotoxic response. Cytotoxic response, it's like killing the pathogen with a sword. So there are two pathways, perforin and granzyme, right? Perforin and granzyme, similar to the names you have already heard in the last hour. So they insert it, and the granzymes enter and they fragment the nuclear material of the organism. So the same way. Second one is a fast ligand pathway. So this is also called as death ligand or CD95. The T cells normally have this fast ligand, a certain protein which can bind to the fast receptor. So when an organism infects a cell, I'm talking of an intracellular organism. So in that process of infection, that surface molecules are all altered, right? And if a fast receptor is expressed, so that can be also recognized by a cytotoxic T lymphocyte, and the process of cellular killing can be started, right? So these are the two ways, but the major way is perforin and granzyme pathway. So this is cytotoxic T cell response. Humoral response, humoral word comes from soluble. So I'm trying to emphasize this point from the outset. So antibodies are important here. So B cells, B lymphocytes, once they're activated, they form plasma cells and T lymphocytes, so memory cells. So lymphocytes may generally, before exposure, the moment they have just come out of their places of development, thymus or bone marrow, they are referred to with the word naive lymphocytes, which have not been exposed to anything, right? Once they are exposed, once they recognize, then they differentiate further into two, effector cells and memory cells. Memory cells stay there, retaining the memory. Effector cells, पिछले केस में था cytotoxic T cell. For a B cell, the effector cell is a plasma cell, activated B lymphocyte. And they release this immunoglobulin. Now this could be bacteria, it could also be toxins. Now these antibodies which are released, they can perform the function of killing with either of these ways. It can neutralize. Say we are talking of diphtheria toxin, for example. Toxin can be neutralized by a protein, antitoxin. So this is an example of neutralization. And antitoxin is also a protein, antibody, which is secreted by the activated B cell or plasma cell. So that's why this is a humoral response. Opsonization, we have already talked. चाहे वो complement के split parts हो या antibodies हो, they can coat, they can bind to the antigens, coat the antigens, and they can make it tastier for the phagocyte to engulf, tastier for the immune response to mount the response. Opsonization. Activation of complement. So antigen antibody complexes, they can activate the complement system also. Complement is a, which one? It is a, you read about complement in the innate system also. So complement is actually, you can consider it as a connecting link between innate immunity and adaptive immunity. दोनों में है ये, right? And antibody dependent cell mediated cytotoxicity. So certain antibodies can also activate natural killer cells. And the natural killer cells can perform the normal function, but because this here, it is possible because of antibody coating. So उसी चीज़ को we are labeling it as antibody dependent cell mediated cytotoxicity, right? They can do it by themselves, but then we'll call it innate response, altered MHC1, right? But if they're recognizing antibody-coated cell, we'll call it ADCC, antibody-dependent cytotoxicity. Now, B lymphocyte response ke liye, for it to flourish and for it to be very effective, what is very important is the Th2 cytokine profile. So, what is that? So, jo other subset of the T cells was the helper cell. Helper cells दो तरीके के, Th1 and Th2, and इनके functions बहुत अलग है. Now, whichever be the case, when a certain response is happening, उनका एक अपना cytokine micro environment develop होता है. So Th2 cytokine environment is important for the antibody mediated or the B cell responses to be very effective. B cell response may we also come across these two terms, clonal selection and clonal expansion. So remember, B cell receptors are somatically generated receptors. So randomly gene rearrangement ke through, there will be a lot of B cells, so each B cell having a very specific type of receptor. So all these things are already present in the body. So one of those B cells, if it is not binding properly with the antigen, वैसे जितने B cells हैं, they will be deleted, they will not be selected. And the one which is very compatible, antigen के साथ एकदम perfectly fit बैठ रहा है जिसका receptor, so that clone of B cells will be selected, the word clonal selection. So in this example, you can see the one with the purple color and one with the yellow color, they are, the antigenic binding sites are not proper. It is a schematic depiction, but the basic idea is कि जितने भी receptor के varieties हैं, जो receptor बहुत अच्छे से पहचानेगा, that particular clone of B cells are selected, and that then undergoes lot of cell division, right? Mitosis, producing clones of that particular selected B cell. So this is called as clonal expansion, clonal selection and clonal expansion. Now remember also, each of the sites, in the beginning we talked of antigens and epitopes. So एक antigen में या एक bacteria for example, it can have lot of epitopes. यानी each of these small epitopes or patterns can be recognized by receptor and different set of receptor and उसका अपना antibody बन सकता है, right? So this is called as a polyclonal response. And why is it important? एक ही organism को, if there are multiple epitopes of the same organism, each epitope will be recognized by a specific B lymphocyte, right? That specific clone will be expanded, उसके प्लाज्मा सेल्स बनेंगे, उसके अपने इम्यूनोग्लोबुलिन्स बनेंगे, राइट? सो ऐसे अगर बहुत सारे क्लोन्स सिलेक्ट हो तो बहुत सारे प्लाज्मा सेल्स विल बी फॉर्म्ड, ईच एक्टिंग ऑन डिफरेंट एस्पेक्ट्स और डिफरेंट एरियाज ऑफ द सेम ऑर्गेनिज्म. सो दिस इज कॉल्ड एज अ पॉलीक्लोनल रिस्पॉन्स. द रिजल्टिंग सीरम एंटीबॉडीज आर हेटेरोजीनस, but they are all directed against the specific organism or antigen. So each one for each epitope, right? Is this point clear? If you attack it from various angles, the effect will be better. The basic idea is this. So this is a schematic depiction कि bacterium को you have these are all very different epitopes, अलग अलग antibodies से एक ही चीज़ को अटैक किया गया, so the effect will definitely be better. Coming to immunoglobulins. So immunoglobulins are the antibodies which are produced by the B lymphocytes, and this immunoglobulins, they have a very specific structure, right? They have a light chain and heavy chain. You have already read about this in biochemistry. I'll rush through this subpart. The light chain has a lesser molecular weight, hence the name light, and heavy one is heavier, hence the name heavy chain. What is important is the first 110 amino acids, this part of both the light and the heavy chain, they vary greatly. So immunoglobulin, so पिछले class में we have talked that B cell receptor जो है वो specifically it is a membrane bound immunoglobulin, right? So it should be able to recognize and bind to the antigen, pathogen, right? So this is where the variety is present. The light, the 110 amino acids of both the light and the heavy chain, this is we call the variable domain of the immunoglobulin. So this is the antigen binding domain, because usme variety hai. And this part is constant, right? This is responsible for the biological activity of the immunoglobulin. Biological activity kaun sa hai? Constant hai. What do we refer as the biological activity? Because some of them should bind to the cell membrane of B cells and they should be functioning as receptors. Some of them should be neutralizing. Some of them should be serving the function of opsonization. तो ये जो उसका specific effector mechanism है, that comes from the heavy, the constant part. But जो light part है, the variable part of both the light and heavy chain, इनका function है to recognize the antigen. So immunoglobulin has these two broad domains, antigen binding domain and a biological activity domain. So a 3D view. So this is immunoglobulin G. So how many immunoglobulins are there? 5. Very good. So you have immunoglobulin G, right? Immunoglobulin A, immunoglobulin M, immunoglobulin D, and immunoglobulin E. Largest among them is the IgM. Why? Because basic structure to yahi hai. It is like a Y, but it is present together as a composite unit or pentamer of 5, right? So that's why size is larger. Now, biochemistry mein you have read about a lot of details, but functional point of view say, for the body's function, these are the points you should not forget, right? So IgG, so you should remember this because this is the antibody or immunoglobulin which can cross the placenta. So if mother is having a certain infection and if eventually the first response of course is by IgM, but eventually the IgM is replaced by IgG. For all acute infection, first response, the first antibody that is produced is an IgM antibody, specific. Later on, the body will produce IgG antibodies to the same organism. So IgM is present in the acute phase, but after that, IgG specific to the antigen is present in the body. So if a mother has some form of infection during pregnancy, so this IgG antibodies can cross the placenta, right? Secondary response, we call them the antibody of secondary response. Primary response, what do you have? IgM. Here we have the secondary response, IgG. IgA, this is the antibody which is present in secretions of the body, like breast milk, tears, saliva, all of this. And this immunoglobulin guards the mucosal surfaces. Respiratory ho, GI tract ho, it is guarding all the mucosal surfaces. So this is also called as the immunoglobulin of mucosa, right? So IgM is the antibody or immunoglobulin of primary response, and it activates the complement system very strongly, right? And also remember, because of its large size, it cannot cross the placenta. Okay. IgD is localized to the membrane of the B cells, and hence it will function as a receptor, B cell receptor. And IgE, these are also localized to the receptors, but these are participating in mast cell degranulation and allergic responses. Okay. Now moving to the helper response. So helper response helps both the cytotoxic response जो भी है and the humoral response, but it does not have its own different way of acting. So hence the name helper. It helps the other two ways of immune response. Helper response activate होने के लिए antigen has to be presented by MHC2, and through the MHC2 it will be presented to which CD4 cell, CD4 or CD8. MHC2 से वो कहाँ पहुँचेगा? MHC is present on the APC dendritic cell, so APC will take it to which cell? CD4 positive cell or CD8 positive cell? CD4 positive cell. So product should be 8. 4 into 2, 8. Remember it this way. Okay. Now, depending again on the organism, और वो किस तरीके से आया है, कि MHC1 से MHC2 से, the response is actually majorly decided at that level कि किस तरीके से वो होगा response. So it will either differentiate into TH1 to helper cell, either वो TH1 में होगा differentiate, either TH2, and for each of these cytokine environments are different. And as I just said, they do not act directly, they enhance the activity of either the cytotoxic response or the humoral response. So look at this diagram. So here it has been presented to CD4 positive cells and TH1 has been activated, right? So TH1 cytokine environment, when I'm referring, so I'm referring to this particular interleukins, interleukin 2, interleukin 3, interferon gamma and T1 necrosis factor alpha. Remember, ये वही cytokines हैं which are already released by the phagocytes, है ना, during the process of battle. Innate में जब ये deal हो रहा है एंटीजन के साथ, तो एक environment बन जाता है. So this environment when it is present, इस level पे it is helping the TH1 to develop further. Why TH1? You look at the effects of TH1. TH1 induces a cytotoxic response, right? Opsonization को बढ़ा रहा है वो. So opsonization will further enhance phagocytosis, right? And it activates the macrophages. At the same time, it is also inhibiting TH2 response. तो body में या तो TH1 response होगा या TH2 होगा. We talk of the helper because there is a mutual inhibition. तो ये environment जब रहेगा, the helper cell will differentiate in line of TH1 and it will further enhance the cytotoxic response. कैसे? Directly it will enhance the cytotoxic T lymphocyte response and it will macrophage ko it will activate and it will enhance the process of opsonization for the process of phagocytosis. If this is the cytokine environment, IL-3, 4, 5, 6, 10, 13, you can actually write down some of this. IL-3, 4, 5, same interleukins, these are for TH2. So if this is the case, TH2 response will be selected, right? And TH2, जब वो बनेगा, so it will increase the activity of the humoral response. B cell का जो activity उसको वो help करेगा. And along with that, it will activate eosinophils and mast cells. So if you go back to your chapter on hematopoiesis, जब वो bone marrow में बन रहा है, तो from the development of the WBCs, you will come across this same interleukins, 3, 4, 5, which is helping the activity of the eosinophil series, basophil series, ये वही है, right? So from here, what do you conclude from this slide? कि helper response कौन सा होगा? That depends on the cytokine environment, right? If it is a TH1 response, it will further activate phagocytosis and cytotoxic response. If it is a TH2 differentiation, it will activate the B cell response, it will activate eosinophils and basophils. Okay. So moving on to the last topic of immunity. So he is very happy, last topic. So we're tolerance. What an irony, right? He is tolerating. So we are going to talk about tolerance now. So immunological tolerance. This is the process or property by which antigen response is not mounted, right? Immune response is not mounted. So technically, if you tend to define it, this is the absence of activation of pathogenic reactivity. बहुत complicated सा definition है. But in simple language, when the immune system is not reacting or not producing antibody response, that is called as a tolerance. So failure of the immune system to respond to an antigen. So is it good or bad? Should we tolerate or not? And is a tolerance mechanism already there in the body? So let us see. So when this tolerance mechanism do not function, it leads to autoimmune diseases, right? That is why it is important. Inappropriate response of the system, immune system against body's own tissues, right? So this is what you call as autoimmune disease. So this just tells us that there is already some level of tolerance in the body. The body's immune system is not acting against the body's own proteins or own tissues. This is a very important point. What are the mechanisms? Yes. So tolerance can be at the level of central, bone marrow या thymus में. It can be at the level of the peripheries, blood में भी हो सकता है. Central level पे what happens is there is a process of clonal deletion. Clonal deletion in the bone marrow and thymus. B cell में we have seen about clonal selection. Clonal selection में a certain type was selected. A certain type of B cell was selected. तो thymus में जिस जगहों पे ऐसा कोई T cell या B cell T cell मिले जिसमें it is reacting against its body's tissues, those clones are deleted. So that is called as clonal deletion. So here what you should remember here is there is some type of education, some type of schooling to this developing B and T lymphocytes. So I'll show you a picture. So this is what happens within the thymus. All of us know by this time that T lymphocyte bone marrow के बाद it is also developing in the thymus. As it goes through the thymus during its development, thymic epithelial cells, recall that they are also APCs, antigen-presenting cells. So वो बारी they bring about, they expose the different antigens or tissues of the body. Body के जो अपने proteins हैं, the thymic epithelial cells are presenting them to the developing T lymphocyte. Why? Developmental phase में the T lymphocyte knows that this is my own body. That's why it is not supposed to react. इसी दौरान, if there are any T lymphocyte which is reacting against the self tissue in the this thymus, those clones are deleted. तो इसमें दो processes हो रहे हैं. First process is positive selection. What is that? Positive selection means only those developing T lymphocytes which are MHC bound or which are recognizing MHCs and MHC bound antigen or proteins, सिर्फ वही select हो रहे हैं. If it is not recognizing a MHC or then it is of no use. The first process that happens is positive selection. If these are not recognizing MHCs only, it is not going to be of any use. So good ones are selected, right? Out of this subset, next, those T cells, developing T cells, which are reacting against the body's own proteins, then they are not selected, negative selection. So thymus में, these are the two important, उनके curriculum में यही है, एक positive selection, एक negative selection. But it's important है tolerance के लिए. Now B cell, it is not developed in the thymus, right? But B cell maybe the same process is there, right? The ones B cell receptors, the ones which are reacting to the body's tissues, they are not selected. And in the B cell, there is one additional process that happens is receptor editing. क्योंकि B cell में MHC वाला point नहीं है because it is not bound to a like MHC thing. But receptors are membrane-bound immunoglobulins. Some receptors, if you catch hold of them in the developing phase, so they are edited again at the gene level, and the receptors are modified so that that B cell is not reacting to the body's own tissue. So this is called as receptor editing. So clonal deletion, positive selection, first the negative selection for T cell, receptor editing and clonal deletion दोनों for B cells. So this is the central mechanism of tolerance. Peripheral mechanisms में दो चीज़ें हैं, suppressor cell and regulatory, which are also called as regulatory cell. And as the name tells you, whenever it finds that the immune response is getting too much, it will suppress it. It will suppress the effector cells by inducing apoptosis. Energy. So energy is where the signal 2 is not delivered. So, एकदम beginning में I talked about when I was talking about the receptors. I told you that receptor में दो signals हो रहे हैं. First signal is the recognition and the second signal is activation. तो ये signal 2 है, if that is not there, that will also lead to loss of tolerance. So these are happening at the level of peripheries. So this is how the body is tolerating. So these are mechanisms of tolerance. If there is a breach of tolerance, किसी भी level पे any of these mechanisms are not functioning, so body will start reacting against its own tissues. I'll show you some examples of that. So this is where you will have autoimmune diseases, right? So first one, that is molecular mimicry. So this is what we see in rheumatic heart disease. It is caused by a streptococcal bacteria, and body is not able to, actually the organism is so clever that the structure of its antigens, it resembles the tissues of the body. So in the process of the body mounting a response against the organism, it also ends up targeting some of its own tissues, the heart for example, right? So here the name of this mechanism is molecular mimicry because of the resemblance. Second one is sequestered antigens, right? The best example of that is during pregnancy. So when a zygote is formed, the zygote is formed in the mother's body. Zygote has the genetic material half of the mother and half of the father, right? But this half, to the mother's body's immune system, this paternal half of the genetic material is foreign, right? And there should be a response, because foreign ke against response hona chahiye. But that does not happen because of the blood placental barrier. One reason is this. So uterine cavity itself is a very privileged site, immunologically privileged site, because immune cells are not able to gain access to them. So because it is not exposed, that is the idea here. See, one very important reason also is जो zygote जब develop हो रहा है trophoblastic stage पे, the other cells are developing. So in the initial stages, the MHCs that are expressed by this developing trophoblastic cells, these are non-classical MHCs. तो MHC 1, 2, 3 तो classical थे, but कुछ ऐसे non-classical MHCs भी है, and because they are non-classical, they are not recognized by the mother's body. There are two reasons why the body is not mounting a response against that. And sometimes we could just be more susceptible because of our genetics, right? Because of our MHCs, which are MHC में कोई mutation हो, so it will make us more susceptible. So this is what is called as genetic susceptibility. So this disease that you see here is the full form is a very big one, systemic lupus erythematosus. Right? You read about this in pathology in details. First year में बस आपको idea होना चाहिए कि these are the examples of autoimmune diseases. Other examples being rheumatoid arthritis, right? Myasthenia gravis. You'll read about this Graves disease, endocrine disease है ये, thyroid का. Hashimoto's thyroiditis, type 1 diabetes mellitus, right? Pancreas के against है, and post-streptococcal glomerulonephritis. Sore throat, which is very common, a bacterial sore throat, but that ends up, the sequelae ends up in the kidney. So there is a inflammatory response in the kidney, post-streptococcal glomerulonephritis. So glomerular membrane is affected. Immunodeficiency syndromes. So here, a different aspect again, when our body's immune system is not self-sufficient, right? So there could be genetic or acquired causes, right? So suppose the phagocytes, they are deficient somehow. It is disease. It is a disease of the phagocytes. Innate का defect है ये. Complement में अगर defect है, complement system में, so we tend to have lot of pus-forming infections, bacterial infections. And adaptive immune system के specifically, again there are lists, you'll find many in the book. AIDS is one example, right? Acquired immunodeficiency syndrome caused by HIV virus, and HIV virus has a specific affinity towards CD4 positive T lymphocytes. So imagine this. हेल्पर सेल का अपना कोई मतलब स्पेसिफिक मेकानिज्म नहीं है. It is helper cell does not have its own distinct set of killing mechanism. It augments the cytotoxic and the humoral response. But that is what this virus is targeting. And helper response, when it is blunted, as in HIV, consequences are devastating, right? There are a lot of opportunistic infections, there are cancers in the body. DJOT syndrome is a syndrome where the thymus gland undergoes aplasia. It is not developing properly. So, in the absence of a proper thymus, T cells will not develop correctly, right? So, with this, I'll stop today. So, immunity mein what is left is something about your allergic reactions. So, that will be covered in the coming classes. All right?